Hello everybody to another wonderful biology video, which I know we all just adore spending our time watching. Um, Hello IB juniors that may or may not be watching this. It's nice to see you. Hopefully this is helpful. I've done a few of these and sometimes they are, I think, helpful to me at least. So this is the review for the second test, which is going to be on some cell theory stuff, apparently. I don't know if the juniors are getting that as a part of their test. I hope not, for their sake. But for us, it's going to have some like cell theory type stuff and like spontaneous generations thrown in there. And then we move into like osmosis and membrane transport. And then we go over to microscopes. I don't really know how she picked these topics, but it's fine. We're going to review them all today. And as usual, I would recommend watching this video on like 1.5 times speed or 2 times speed because I know that I can only talk so quickly. I do tend to talk somewhat fast, but like there's a lot of content and I'm really hoping this doesn't take too terribly long because it's already like 8 p.m. But I'm just going to go ahead and get started and hopefully this is helpful. So the first statement that we have for this test is that all organisms maintain life by carrying out common functions. And then it asks us to list the common functions carried out by all life. And I have those sitting next to me and they're also up on the screen. Metabolism, growth, reproduction, response, homeostasis, nutrition, excretion. And that is on page 5 of our textbook. And she also mentioned made of cells, but that's definitely not a function, that's a property. But if it asks, that is also technically a commonality of all organisms, is that they are made of cells. But this is something you're going to want to need to memorize, so I'm going to repeat it a few times, and maybe you'll repeat it with me. And I'll read off of the screen this time so we can all read together. Metabolism, homeostasis, excretion, growth, development, adaptation, evolution. Metabolism, homeostasis, excretion, growth, development, adaptation, evolution. Metabolism, homeostasis, excretion, growth, development, adaptation, evolution. I will probably repeat that a few times before the test to myself as well, but, to each their own. And so now I'm going to just briefly, like in my own words, I'm not reading off of a textbook or anything here, I'm going to define these words because it asked us to do that. So metabolism is, I don't even know this is the first one Metabolism includes all the chemical reactions that occur within an organism. So that includes converting energy from one form to another. So metabolism is just chemical reactions. Homeostasis is the ability to keep equilibrium in the body. So that could be something like pertaining to keeping stable temperature. Excretion is excretion, growth is growth. Development is just kind of like a slightly different than growth. Growth could just mean exclusively in size, whereas development could be changes in a single organism, whereas adaptation and evolution refer more to like over a long period of time. Adaptation referring to, of course, adapting to the environment, changing, so that it can be better suited to where it lives, and evolution is that change over a long period of time. So next this says, state why viruses are not typically considered to be alive. We did not go over this in the review in class, but I will go over it very briefly for us today. Viruses aren't considered to be alive because they don't do any of those things. I mean, some of them, but like, they're not made of cells. They don't really adapt to their environment necessarily, they don't develop or grow, they don't excrete certainly, and they probably don't really maintain homeostasis, but the main thing that you can use is that they're not made of cells, and all living things are made of cells. Comparing sexual and asexual reproduction is another thing that we did not go over in class, but I will go over them very, very briefly. Just obviously sexual reproduction is going to be two animals or organisms that actively mate, and they have a female and a male part, and this could be flowers as well, they also do. And sexual reproduction can do sexual reproduction, but they have the male and female, and both are needed, whereas asexual reproduction doesn't necessarily include that. An example of asexual reproduction would be like pollen is an example of asexual reproduction, I believe. But that's kind of the difference is sex. Comparing life, responding and adapting to the environment. So responding to the environment would just be like, there's a big fish over there, I'm going to go eat it. Whereas adapting to the environment might be like, oh, it's hot, I'm going to cool myself down. So that's kind of the difference there, I think, a little bit. Again, we didn't go over that in class either. And then finally on this list is comparing how autotrophs and heterotrophs obtain energy. This is another thing we did not go over in class. I hope we all know this by now, but autotrophs are going to be the ones that make their own food, whereas heterotrophs are the ones that eat other organisms for food. Moving right along, unicellular organisms such as paramecium, and that word that I can't read, perform all functions of life. Now let's switch over to this tab, and we are going to look at the paramecium first. And so these are, it listed out, hopefully, all of the things of life, and I'm just going to read through them very briefly, and we can note the differences between the two, because that might be the easiest way to do this. Excretion is the plasma membrane controlling entry and exit. Metabolism is that there are metabolic pathways in the cytoplasm. Nutrition is that food vacuoles contain organisms that the paramecium has consumed. So there are many vacuoles, because then if we move up to the top here with homeostasis, there are contractile vacuole fill up with water and expel through this plasma membrane that deals with excretion, and that manages the water content. So there are several different kinds of vacuoles going on here, and they kind of relate to this excretion situation. But I feel like that's one of the more important things of paramecium, because if I remember correctly, the other one does not have this homeostasis surrounding. Well, I guess it does, and she just told us wrong in class. Great. Response is going to be that the cilia move the paramecium in response to changes in the environment. So if there's food, we're going to move towards it with those cilia. We're just going to walk ourselves over there. I mean, that's true of everything, but apparently it's also true of paramecium, so that's good to know. Reproduction is that the nucleus can divide and support cell division by mitosis. Reproduction is often asexual, there's that word again, with the paramecium. And finally, growth. After consuming and assimilating biomass from food, the paramecium will get larger until it divides. And again, that kind of deals with reproduction. It's going to get larger and then it divides, and that's the reproduction. And bada bing, bada boom, there you go. So I guess take of that what you will. It's pretty straightforward, I think. I guess the only thing that we could really pull out that's specifically important is the plasma membrane and the food vacuoles versus the contractile vacuoles with the water and the expulsion. Moving right along to the chlorella, if that's how you say that, which we're going to pretend it is. Excretion, again, there's that plasma membrane that's dealing with waste oxygen. Keep in mind, this was metabolic waste for the paramecium, but down here for the chlorella, it's going to be waste oxygen. Metabolism is the same as before, metabolic pathways in the cytoplasm. Nutrition is also going to be different. The chlorella, you might notice in this gorgeous picture here, is green, which means that photosynthesis is happening. And that helps provide the algae with food. So nutrition is going to be different from the paramecium as well, because the chlorella does that nifty little thing called photosynthesis, whereas the paramecium had this little cilia that it would walk on up to a food source and store it in those vacuoles. Growth. After consuming and assimilating biomass from food, the algae will get larger until it divides. This is, again, a connection between the paramecium because it also got larger until it divides. In reproduction, the nucleus can divide and mitosis happens, and the cells in this picture undergoing cytokinesis. It's all fun stuff, like same as before, really. Get bigger. divide. Good stuff, good stuff. Response. The wave of action of the cilia moves the algae in response to changes in the environment towards light. So this is, again, similar to the paramecium, except the paramecium was moving towards food, and this would move towards light. Homeostasis is, once again, those contractile vacuoles filling up with water and expelling it through the plasma membrane. So the really big difference to note here that I'm getting is that the chlorella is going to have this waste oxygen, whereas the paramecium is going to have this metabolic waste. And then obviously the chlorella is doing photosynthesis, so it's going to go towards the light, whereas the paramecium was gobbling up other organisms, so it had those food vacuoles, and it moved in response to other organisms. Moving right along, advances in the scientific understanding led to the development of the cell theory, one of the basic principles of biology. So let's state the 3 parts of the cell theory. And again, I have this open to page 4 of my textbook, and I'm going to read right from there for these. Number 1, all organisms are comprised of 1 or more cells. And did you know that? I knew that. But now you know it's a part of the cell theory. Isn't that fun? So yes, everything is made of cells. Well, everything that's an organism is made of cells, unlike viruses. Number 2, cells are the smallest units of life. There is no evidence suggesting that a smaller unit of life exists. Everything that we found that can be considered an organism has at least 1 cell. And 3, all cells come from pre-existing cells. So this means that there's some dividing action, the nucleus is splitting, and we are undergoing mitosis. Some good stuff like that is happening. So then it wants us to kind of outline the evidence that supports the cell theory. Robert Hooke, and you can think of him as the cell pirate, I guess, if you need to. Robert Hooke here, because he's got a hook. Aren't I hilarious? He described cells in 1665 because he looked at them with a self-built microscope. Go him. We're going to talk about microscopes just here in a bit, so that's another way to remember this. And then a different guy observed the cells and referred to them as little animals, and then a botanist called the plants made up of independent square beings called cells. And then we've just kind of gone from there where it's pretty clear that everything's made of cells, yeah? Second principle continues to gain support today because no one has been able to find any living entity that is not made up of at least 1 cell. So that's a really easy one to remember. Everything's made of cells, and the cells are the smallest building block of life because no one can prove otherwise. And finally, we move on to all cells come from pre-existing cells, which has to do with spontaneous generation, which is actually one of my favorite topics. So let's talk about spontaneous generation really quick. So this guy, Louis Pasteur, of course, he proved that spontaneous generation was not a thing. So what is spontaneous generation, and why is it important that we know it's not a thing? So spontaneous generation was this idea that beings, organisms, they just existed. They just came into being. They just popped up. They didn't come from pre-existing cells. That. Yeah? So Louis Pasteur said, no. That's not how this works. This spontaneous generation business is not true. And so what he did is he did a series of these swan neck broth experiments surrounding decomposition. So he did like 3 trials that are important. Each of them started with applying heat to this broth that killed all the bacteria and all the cells and all the things that would be alive. And first he applied heat and just let the flask sit. And I don't have it up here, unfortunately. But this did show that, things couldn't get in through the swan neck. Maybe these cells aren't just popping into existence. And so to prove that this mixture of his could produce cells, he did the same thing where he applied the heat. Then he removed this neck that was preventing the organisms from getting in. let it sit, and bada bing, bada boom, would you imagine there's bacteria now? And then finally, he messed with the swan neck itself, applied this heat, tilted the flask sideways into the swan neck, so it's getting into where this the bacteria will eventually sit. And then he kind of noticed that while there's nothing in the mixture, because the organisms wouldn't be able to get into the actual broth mixture, there were some in the swan neck where he had previously had the broth. And so this kind of proved that no, they don't just come out of nowhere. Yes, they can come into existence, and that they have to come from the outside world where there is pre-existing cells. And this kind of proved that pre-existing cells were needed to establish life, or actual cells, new cells, quote unquote, in this broth mixture. Moving right along from there, we are going to look at the differences. Wow. Although living organisms are composed of cells that share common characteristics, there are discrepancies from the typical. So there are basically 3 examples here that we need to know. And unfortunately, a lot of this does just come down to straight up memorizing, which really sucks. But we do have these gorgeous pictures here that we can talk about and kind of compare. So muscle cells, which we have in our very own body, aren't we lucky? They have multinucleated fibers. Well, what is a smaller word for that? Their cells have more than one nucleus, which is not the norm. We usually think of cells having one single nucleus, but not in muscle cells. They have long multinucleic fibers. So they're just really long cells with lots of nuclei. A septate hyphae, maybe, have no cellular partitions. So in this picture here, we are looking at several cells in this straight line, but unlike the muscle fibers where it is a long cell with many nuclei, this is many separate cells that don't have any like cell walls or cell membranes, and they all just kind of puddle themselves together. And then finally, the giant algae can be very large. So we usually think of cells being the smallest building block of life. Well, they can be pretty big too, even greater than 70 millimeters, which is huge. Like, get out a ruler and look at that. That's a big stinking cell. Like, that's okay. But that's what makes them an atypical cells. The muscle cells have multi-nucleus. The fungal hyphae, that one, it doesn't have any cell partitions, and the giant algae is big. Cell surface to volume is an important limitation to cell size. So, what we need to know for this is that we need to outline the activities occurring in the volume and at the surface of the cell, calculate each of these, explain the benefits and limitations of using cube models to model the surface area and volume, describe the relationship between cell size and the surface area volume ratio of the cell. Explain why cells are often limited by size by the surface area volume ratio. And list 3 adaptations of cells that can maximize the surface area volume ratio. So we certainly did not go over much of this in class this year for my IB seniors, and for my IB juniors that might be watching, I don't know if you guys went over this at all. Aren't you just the luckiest? I went over that. So here we have this beautiful website that I found that I'm basically just going to read from because I don't know. I could go into my old notes, and I probably will do this, but for the sake of recording this and being somewhat helpful, I'm not going to do that. And so what I'll do instead is we can just read over this video, or read over this article together. And you can see I kind of scrolled to the middle to where I think it's like appropriate or whatever. And so smaller single-celled organisms have a higher surface area to volume ratio, which allows them to rely on oxygen and material diffusing into the cell and waste diffusing out in order to survive. So let's break that sentence down really fast. So small single-celled organisms, so we can think back to our paramecium or whatever we want, have a high surface area, which means they have a lot of surface area, and little volume. So if we look at this guy here, we just imagine this is our cell. This circle, even if the smaller we made it, it would have a lot more surface area and a lot less volume. Whereas if we had a bigger cell, that would be the opposite. If we had a big circle, it would have a lot more volume when compared to the surface area, right? So this allows materials to diffuse in and out of the cell, and that helps it survive. The higher the surface area to volume ratio they have, so that means the more surface area, like those smaller cells, to volume, again, these high surface area, low volume would be these single-celled organisms that are small. Probably excluding that algae from earlier and things like that. The more effective this process can be. So the more surface area, the more surface area when compared to the volume. So let's go over to this. I think I can do this better. So if there is a small cell and a big cell, we can look at this like here, it's supporting the same amount of life, right? Because this is alive and this is alive. We're all alive here, I hope. I'm not. So this would be supporting life, both of them, theoretically, right? Yes. But with this, there's so little volume to the surface area that things are able to diffuse in and out of it much easier. Whereas this, it would have to travel a long winding path and it might get a little lost or whatever. It's harder to get in when there's a lot more volume. And this just means that things can diffuse in and out much easier. So larger animals that require specialized organs that effectively increase the surface area available for exchange processes, a circulatory system to move material and heat energy. So that would be, those larger animals would require those specialized organs because they don't have as good diffusion rates, right? So increased volume can lead to biological problems, like apparently this fictional giant gorilla that would have had insufficient lung surface area to meet his oxygen needs. Great. For small organisms with their high area to volume ratio, friction and fluid dynamics are relatively much more important, and gravity is much less important compared to larger animals. So if we flip back over to here, because I think that's about all we're going to get out of the website. We didn't calculate surface area to volume, but I think we can all handle that. It's just a little bit of dividing, probably. The benefits and limitations to using cube models. Cubes are going to have a much more stable, a much more linear looking surface area to volume ratio compared to a cell, which can be many shapes. That's a limitation. But a benefit is that it does give us an idea of what this surface area to volume ratio would look like. It's just not a perfect depiction. We kind of did describe the relationship between cell size and surface area volume. Why are cells limited in size? We got there too. It's because at a certain point, you're not going to be able to function as well because things aren't going to be able to diffuse in and out of the cell as easily. And then 3 adaptations of cells that maximize the surface area volume ratio. I can only remember one from last year, and that was the red blood cell, which if we remember is kind of dimpled. It's kind of like a disc if you pushed your thumb in and dimpled it, so it has a little bit of a dip. And this allows there to be more surface area and still basically the same amount of volume, right? Moving right along. Particles can move across membranes by simple diffusion, facilitated diffusion, osmosis, and active transport. Woo! So I did a whole 20-ish minute, it might even be 12 minutes, I don't remember. I did a whole video on this like 3 weeks ago. And I really, really recommend that if you have major questions, go watch that video because I talked about this in much more depth. I did really nice drawings, like it was a good time. So I'm not going to go through this in as much detail just because I don't feel like I need to, but I will go through these points very quickly. Simple diffusion is going to be that high to low. It's along the gradient. It's small particles, non-charged particles. Two examples of molecules would be like oxygen or glucose, I'm guessing, in and out of cells. It's waste, it's energy, it's all the good stuff. Yeah, that's not charged and it's very small and it's easy to get through. Factors that could regulate the rate of diffusion would be the fluidity of the membrane, the gradient, theoretically. Facilitated diffusion is going to be diffusion that requires a protein helper, and that is still along the gradient. However, this time, the particles might be slightly larger, or they might be charged, meaning that they require this protein helper to help them get along the membrane. But again, it's still along the gradient, which means it's high to low. One example of facilitated diffusion along a protein channel is probably going to be the one that I can't think of right now that we talked about at some point in our lives. It's fine. I mean, you can look it up. You have fingers. I trust you. Define osmosis. Osmosis is going to be diffusion along the gradient with regards to water. So what is going to be moving in this situation is water. The solutes, which is like the sugar or salt or whatever that the water is like needing to pair up with basically, is going to stay where it is. But the water is going to move from place to place. The direction of water movement based upon differences in solute concentration. If there is more solute in one place and less solute in another, the water will move from where there is less solute to where there is more solute. So this could mean that cells are going to be put in solutions that are hypotonic and they will swell because they have more solutes. If this same cell is put in a solution that is hypertonic, it will shrink because the water will go out into the solution that has more solutes. Isotonic solutions will always result in zero net movement. Active transport and passive transport are different because active transport requires ATP and passive transport does not. Active transport is going to be against the gradient, meaning low to high, whereas passive transport is always going to be along the gradient, meaning high to low. Explain one example of active transport of molecules in and out of the cells using... you have fingers. You can Google that. I'm not explaining that. You can do it. I promise. Which probably means I'll get that question wrong on the test. Because I will forget. Estimation of osmolarity in tissues by bathing samples in hypotonic and hypertonic solutions. So we went over all these words already, so I'm not going to go over them again. A lot of this is just her going over, I'm guessing, what we did in that lab that I know everybody did, where we did the potatoes and the water and yeah. So I think you can all handle the mean value of a data set, and this, like we don't need to hand estimate the standard deviation. It is kind of important to know that the standard deviation is summing up the values of the mean. So it's kind of like the spread of values. So if the values are much tighter, it will have a smaller standard deviation, whereas much bigger is going to be a larger standard deviation. And this is useful because it kind of shows how accurate our data is or how strongly our data supports our point. If you look at two error bars and it's, hold on. If the error bars are like this, then maybe the data isn't as supported, even if the points are in different places. But if they're closer to being like this, then the data probably is in support. I hope that made sense. That was a really rushed drawing because I don't think we're going to need to know that very well. Estimate the type of graph used to represent experimental results. When dealing with osmolarity, we're going to use that scatter plot, and we're going to draw the lines on that scatter plot. Error bars are a graphical representation of the variability of the data. I stated it. Moving on. I don't think we need to go over any of the specific graphing things. Osmolarity of a sample changes when placed in solutions of various toxicities. Going back to this potato plant osmolarity lab we did, when the potato swelled, that means it got positive percentage of mass change. This indicated a time when it was in a hypotonic solution because there were more solutes inside the potato than there were outside. So positive percent is hypotonic. Whereas when it was a negative percent, the water was going out of the potato. It was in a hypertonic solution where there were more solutes outside of the potato in the solution it was in. Where the graph crosses the zero net change line, that is where it is in an isotonic solution because there is not gain and there is not loss. Qualitative and quantitative. Qualitative is going to be like what you can see, if something is green or brown, if something is big or small, whereas quantitative is going to be something that you can measure. So that might be the weight or the content of the solution. The measurement uncertainty. Measurement uncertainty is always going to be you take your smallest unit. So if we're using millimeters, we might have a measurement of 1.2 millimeters. We would take that 0.2, and that would be a smallest measurement of 0.1, and we would divide that by 2. I hope this makes sense. So we would have an uncertainty of 0.05. So for our 1.2 millimeter measurement, there would be an uncertainty of 0.05. And this is because on our theoretical imaginary ruler, we might be 0.5 above or below and just can't measure that because the measurements didn't go that exact on our ruler. The need for repeated measurements in experimental design helps us accurately reflect data, make sure things aren't flukes. Sometimes things just happen once and that's kind of weird. But if they happen multiple times, we can start to assume that it's a trend in data and a trend in which things go down. Controlled variables help us to regulate what could be impacting our results, and this also helps with the repeated measurements in case there are many uncontrolled variables. Tissues or organs to be used in medical procedures must be bathed in the solution with the same osmolarity as the cytoplasm to prevent osmosis. This should just make sense to you, right? Like if we are transferring a heart, we don't want the heart to swell up before we put it in somebody else's body, and we certainly don't want it to shrivel up. So yes, of course, normal saline needs to be used, and same situation with the osmolarity as before. And just in general, isotonic solutions are a big thumbs up. Moving on to, moving on to microscopes, yay! So higher resolution. Resolution is just going to be the clarity. Electron microscopes are going to have a higher resolution than light microscopes. So light microscopes are the kind we have in the classroom, whereas electron microscopes are going to be the kind that we never get to use, but they take such good pictures, and you will always be able to tell. On any question where you're being asked which one is the electron microscope, it is always going to be the clear one, or the one with higher resolution. That's because the electron microscope has a much higher maximum resolution. So it can be up to 2,000 times with light microscopes, which sounds impressive because it is, but electron microscopes, it's 250,000 times. That's a big difference, right? So that's going to be the difference in magnification, and of course the resolution is just going to be better in electron microscopes regardless. Developments in scientific research follows the improvement in apparatus. So that means that when we're able to see things better with this electron microscope than we ever could with the light microscopes we've been using for years, yes, of course we're going to make new developments because you can see more things, you can learn more things with new apparatus. An improvement in apparatus allows for a greater understanding of cell structure in this case, because if you can see the cell clearer and closer up, then you're going to have a better understanding of it. Moving right along. Use of the light microscope to investigate the cell structure in tissues. I'm not going to label the parts of a microscope, and I'm just going to hope that we don't need to. But also, like, it's obvious. You should be able to. You know the arm. The only thing that I think can get even relatively confusing is the fine versus the coarse magnification focuser thingy. And the fine one is always going to be the small one. Magnification is how close up we can see something. The magnification and the ocular... Whenever you are given the ocular lens, which is the one that you look into with your eye, and the objective lens, which is the one you can spin, if you want to find out the total microscope magnification, multiply the two. So it's always going to be O times O, ocular versus objective, right? So if you are given that there is a 10 ocular and a 4 objective, then you are going to have a 40 times magnification in general. Measure the field of view diameter of a microscope under low power. So what we're going to do here is zoom the microscope all the way out on the lowest objective lens, which in our case is 4. And what we're going to do is we're going to take a ruler with millimeters and we're going to put it underneath. And you're going to count how many spaces you see. So it's maybe 4.5. In that little eyepiece, you see 4.5 millimeters. Once you've lined up one of the sides, like, you know how to measure things. Same as always. And so what you're going to do is you're going to count that, and that is going to be your field of view under low power. Calculating field of view under higher power is going to be a little bit different. So here's a good example. Light, electron. Look at how much nicer that is. Isn't that beautiful? Here we go. Here's our field of view. So, okay, this is important to measure. This is important to point out as well. When you have centimeters, to go to millimeters. And so if you have cm and you need to go to mm, it's times 10. If you have mm and you need to go to um, it's times 1,000. Okay? Good stuff. If the power is changing and you need to figure out the field of view, or the field of view on a higher power. So again, for a low power, all you need to do is count how many millimeters you see on a ruler. Shut up, Wesley. But if you have a medium or high power and you need to find the field of view, you take the diameter that you saw before, and again, that's just the count of how many millimeters you saw, times the magnification of the low power magnification. This is the ocular times the objective. This is 40 for us. It might not be 40 on the test, But in our microscopes, it's 40. So if you have 4.3 times 40 divided by the higher objective. So magnification of the objective. Technically, the objective would be 4, and the higher objective might be 10 or 40 or whatever it is. But if you think about it logically for a second, you're going to get the same answer. All right. But that's how you get a field of view in higher. So it would be the diameter that you found before, which is, again, just the count that you got, times the magnification of the low, which for us is always going to be 4 or 40, divided by the magnification of the higher power, which could be somewhere between 40, or just, it could be like 100, it could be 400, it could be whatever. But that's how you go about that. Good stuff. Estimate the size of a sample. I don't think we're going to have this on the test. I believe she said that. However, just for kicks and giggles, let's look at a nice little example here. Really, really fast. Here, if we're measuring this little dude right here, he fits across the screen approximately 10 times. Don't say 10.1. Now you're getting specific. That's a little gutsy of you. I'm not doing that. It's about 10 times. So you're going to take the estimated length, which is 1/10, times that field of view that you had before, which in this case we've determined is 400 UM. 400 times 1/10 is 40. And that's how you're going to get that size. So again, you're approximating how many could fit across on this screen. Like in this one, it would be about 2.3. Does it look like exactly 2.3? No, but we're just saying it's about 2.3. And don't say 2.30 because now you're getting specific. 2.30 is specific. 2.3 is fine. And again, if we had the field of view as 600 UM, we could divide that by 2.3, and then we get about 300, always round, because it is an estimate and it is not a final value. It can only be it must be rounded to one significant digit, which means one number and however many zeros. How to focus a microscope on a sample, like you should know that. Like you just zoom it in and out until you see it, okay? Drawing cell structures as seen with a light microscope. That is going to be on this PowerPoint. You're going to need, I believe it says something like a title and all this fancy stuff. It's somewhere. It's unnecessary. Like nobody needs that in their life. We're not going to need to do this on the test because you're not going to need to do this on the test. But always give it a title. Always make sure you use a scale of how big it would be, like the little scale bar thing, which you see nowhere apparently. Here, this is a scale bar. If you say this is 0.3 mm, obviously this drawing is much bigger than 0.3 mm, but in this case, it's going to be 0.3 mm is the actual thing. To find the magnification on the paper, because of course it would be way too easy to just have it be simple, what you're going to do is measure this with your ruler. Let's say you get 300 mm and then divide that by 0.3 mm. You are not measuring the drawing. You are measuring the scale bar. Because I might think the organism ends about here, but you might think it ends about here, but actually it ends about here. That's not good enough. Measure the scale bar where there is a very clear start to finish. Measure the scale bar, then divide it by whatever the scale bar says. That's going to be your magnification. So in that example that I just gave, I think it would be like 1,000. Fine, right? Good, we're fine. And again, to go from cm, it's times 10. That would get you to mm. To get from mm to um, it's times 1,000. On this really terrible example here, It's showing you exactly what I said. Measured size of the drawing divided by, and then it doesn't show it because it didn't download the PowerPoint, divided by the scale bars thing. And that would get you your magnification. Always round it to one significant digit. This is an estimate. How to draw cell structures with a microscope using sharp, carefully joined lines and straight edges for labels. I really do not think we are going to need to, I don't think we're going to need to draw any cell structures. I don't foresee that being a thing, but just in case, be neat about it, add a title, add your magnification, add your scale bar, add by how much the scale bar actually is. Label your parts. Just like, if you can add more detail, because chances are you're not going to get points off from having too much detail. Micrograph. What's a photograph? What's a microscope? It's a picture with a microscope. I hope you can get the dramatic effect of me leaning into my computer as I say these things, because I don't think you can hear it, but it's happening. State why the magnification of a drawing or micrograph is not the same as the magnification of the microscope. Because if something is 0.3 millimeters, you are not going to draw a drawing of 0.3 millimeters on your paper. Therefore, there is going to be a magnification between the actual thing and the drawing that you're drawing. Use a formula to calculate the magnification. Measure the scale bar. Measured scale bar divided by scale bar what it says. Boom, magnification. Make sure they're in the same units. Make sure they're in the same units. Make sure they're in the same units. Are they in the same units? I hope you check. They should be in the same units. If they're not, fix it. Fix it. Fix it. It's always going to be times some form of 10. If given the magnification of a micrograph or drawing, use a formula to calculate the actual size of a specimen. It's going to be the exact opposite of what we just did. So you might have to measure the thing and then divide it by the magnification. So if we go back to this example that I had going on, if we go back to this example that I had going on here, where I said it was 300 mm and the magnification was 1,000. 300 mm divided by 1,000 is going to be 0.3, and that's a scale bar. And I understand that's a scale bar, but we can pretend it's the measured organism. We can pretend whatever we want. That would be how you determine the actual size of the specimen. Is you're going to determine the magnification, measure the specimen, drawing, the entire thing, not just the scale bar this time, and adjust accordingly, right? Divide by the magnification. So there are 3 parts to this. Determine the magnification, measure the drawing for what the drawing is, divide that number by the magnification. Whew. All right, I think that's everything. I hope that's everything because I'm not going over anything else. I hope this was helpful to you in some way, shape, or form, and if it wasn't, don't tell me because I don't want to know. Good luck on the test. Sorry, IB Juniors, that this is getting out at like 9 p.m. on the day before your test. But IB Seniors, you have like a whole day to watch this. It's like 40 minutes probably. Have fun and good luck on the test. And come to me with any questions. I can give you my Discord handle, IB Juniors. Find me somewhere. I'm around. Ask someone. Ask Haley. Haley knows where I am. Haley can message me. And if you have more questions, go to our teacher. But I'm always open to answer questions or clarify things that I said in this because I know that I'm running on like 0 breaks for like 12 hours. Have a good day. Do good things. That's it.